Welcome to Almost Here, Round the Corner Future Technology podcasts with Richard Jacobs. Future technologies poised to transform our lives for better or worse are the focus of this podcast. Almost here means these technologies are now here and starting to be used, or just around the corner, from Bitcoin to artificial intelligence, 3D printing, blockchain, virtual reality, and more.
Hello, this is Richard Jacobs with the Future Tech Podcast. I have two guests today, Susan Stevenson, Executive Director, and Craig Mickanin, Director at Novartis, Novartis. Novartis is a large, well-established company that is involved in a lot of cell therapies, gene editing, etc. We get a lot to cover. So welcome, Susan and Craig. How are you?
Great, Richard. How are you? Thank you.
Fine, Richard. Thanks.
Great. Well, if you guys can tell me, Novartis, I'm sure, covers many areas. Can you give a brief overview of the company and the different initiatives that are going on right now, and then we'll dive into a couple and get more details.
Great. I'll take that question. So, as you know, Novartis is a global healthcare company based in Switzerland that provides healthcare solutions to address the needs of patients and societies around the world. We use science-based innovations, and we work to address some of society's most challenging healthcare issues. Our portfolio includes innovative pharmaceuticals, oncology medicines, generics, and biosimilars, as well as eye care devices. Craig and I are in the research arm of the company called the Novartis Institutes for BioMedical Research, which is responsible for leading early drug discovery and early R&D for Novartis. We embrace multiple collaborations across disciplines with both biotech companies as well as academic partnerships in order to develop innovative medicines and advance exciting science to patients. Within NIBR, we have about 6,000 associates dedicated to drug discovery. We focus on different disease areas ranging from oncology, cardiometabolic, respiratory, neuroscience, immunology, dermatology, and ophthalmology. Now, that finally gets us down to the area that Craig and I have been working in for many years, and that is the development of cell and gene therapies. This is a really exciting time for cell and gene therapy within Novartis as well as in the entire field. Novartis and NIBR have made this a strategic priority because of the potential to develop transformative medicines for a range of diseases. And so I head up the cell and gene therapy initiative within Novartis, which helps to coordinate and execute a variety of different gene therapy and cell therapy programs for multiple diseases. So I'll stop there.
Okay, well, very good. Within the world of cell and gene therapies, what are some examples of those that are recent that maybe have gotten through clinical trials that people are now going to start seeing when they need medical care? Or are they too early yet and they're still in clinical trials?
No, Novartis has had a fantastic example. The first approved ex vivo autologous T-cell therapy for B-cell lymphoma. The approved product is called Kymriah, which was approved in the summer of last year, 2017. That developed since 2012 in partnership with Carl June at the University of Pennsylvania. And so that is a curative therapy for B-cell lymphoma. So that's a really exciting example and has helped establish all of our internal infrastructure for development of cell-based therapies that we're now applying to other areas where we're developing cell therapies.
So, okay, you used just to clarify for listeners, you said autologous, meaning the person's own cells, ex vivo, so I guess they're cultured outside the body, and it's a T-cell therapy. So can you talk a little bit about the mechanics of what happens in that therapy? You'll culture someone's cells, you'll make more of them outside the body. How will you alter them, and then how will you reintroduce them, and what will that do to combat the lymphoma?
Mm-hmm. So gene therapy in general can be divided into two ways to treat the patient. One in an ex vivo mode, where, as you just described, we take the cells out of the patient, modify them. In the example of the CAR-T program, we use a lentiviral vector to transduce the isolated T cells to introduce permanently in those cells an antibody construct that allows those T cells to be targeted to the tumor type. In this case, it's an antibody against CD19, which targets the B-cell lymphoma. those modified cells are grown in culture after they've been exposed to the lentiviral construct, and then return back to the patient, and then those patients home to the tumor cells in the patients to destroy them. And so that's an example of an ex vivo cell therapy. Again, autologous therapy, meaning the patient's own cells are modified and put back. The other way that we could introduce genes into patients is using viral-based delivery systems where they're injected directly into the patient. And there we have two actually ongoing clinical studies, one using an adenoviral vector, which is injected into the ear, taking advantage of a local delivery setting. The second example was directly injected into the back of the eye to go out to restore and deliver a functional copy of a gene that's mutated in an inherited retinopathy situation. And so those are three Novartis programs. The Kymriah example is approved and it's on the market and it's being sold. The ear and the eye programs are just in early clinical development. In fact, we've just started the first ophthalmology clinical study this year. The ear clinical study has been going on for a few years now, and we're learning a lot from that initial clinical experience. But those, going back to in vivo versus ex vivo, those are some examples of actual active Novartis programs ongoing.
Yeah, it's amazing that you can have a therapy like that. Wow. And there's millions of things to focus on. So how do you guys determine where the focuses are at Novartis? There's so many diseases, so many things to work on.
Well, I think that one, of course, greatest medical need, no available pharmaceutical or other ways to approach the disease. Cell and gene therapy is, I think, more work up front because of it's a different delivery system, a different modality. But in the end, given that it's a single administration potential for curative therapies, it's worth the upfront investment. And so we look at all possible ways that we could, in diseases where we can apply and develop gene therapy, and we have we go through this decision tree and we select those indications where we think we will have the biggest clinical benefit. And restoring blindness is one, restoring hearing is the other. There are no pharmaceutical interventions available. Curing cancer in the case of B-cell lymphoma is another. Novartis just recently acquired Avexis and developing an in vivo gene therapy to go after a genetic neurodevelopmental disorder, spinal muscular atrophy, or SMA. And the clinical data that they're showing is just amazing. I mean, these children should not be living past the age of 1 or 2, and they are gotten significant survival benefit. And that robust clinical data was the basis of Novartis being interested in the acquisition of Avexis this year. And so another example of a transformative medicine that hopefully will be broadly available next year if approved. And so that's where the team is interacting and trying to submit the necessary documentation for approval.
So what's been your experience in this field? Has it been a slow accumulation of therapies and results, or has there been a sudden resurgence or a sudden breakthrough in the past few years that allows these therapies to happen?
Cell and gene therapy has been the field, I think, started back in 1989, 1990. The first gene therapy study was in September 1990. I actually was at the company that did that first. It was a study. It was an ex vivo modified hematopoietic stem cell therapy going after a condition called severe combined immunodeficiency disorder. And again, it was a gene replacement approach with autologous therapies and using a virus to deliver the gene back into stem cells and delivering those modified cells back into the patient. That therapy just got approved in 2016, and it's called the Strimvelis. Mm-hmm. And so it has taken a long time, and I think it's because of the novelty of the technology, wanting to understand the efficacy, but but very importantly, safety. And it's just taken time. There were some setbacks in the entire field in the late 90s, and I think that the whole field had to take a pause. And now that we've understood the delivery platforms a bit better, and we understand better their limitations, we're applying them now in settings where they have a better chance of success. For example, in local administration to the back of the eye or into the ear, we're not thinking that we're going to cure everything. And so there's some lessons learned along the way. And I think now, with 3 new approvals last year in the field with Kymriah and Yescarta and Luxturna, now you're going to be seeing more of these gene therapy medicines coming forward and being approved. And it's just a really exciting time now. It has taken a while to get here, but now I think we're applying and trying to develop gene therapy in more appropriate ways.
Somewhat ironically, both Sue and I have been in this field for well over 20 years. Mm-hmm. Sue worked previously at a Novartis company called GTI, Genetic Therapy Incorporated, down in Maryland. Mm-hmm. In the mid-'90s and late '90s, I was at the University of Pennsylvania, affiliated with the Institute for Human Gene Therapy, and then at a company called Onyx Pharmaceuticals, which was also developing adenoviral approaches for tumor therapeutics. And it was really, I think, some of the early setbacks which put the field on hold for probably around 10 or so years before it took a series of advances in technologies. I think there was a clearer and potentially more conservative regulatory path. And now what we're starting to see is a resurgence in this field. So in some ways, it's kind of coming full circle on things that we were involved in many years ago and always felt had the potential to be transformative in medicine.
Okay, that's what I wanted to ask. Yeah. There's at least a few companies out there. Some of them say, bank your stem cells for future therapies. There's places where you can bank, placental blood, cord blood, things like this, stem cells. What's your opinion of doing something like that in the hopes that in the next, 5, 10, 20, 30 years that your own stem cells will be available for a therapy for you? Do you think it's needed? Do you think, what are your thoughts around that, those kind of therapies?
Well, currently here at Novartis, we're not really engaged in any efforts around true stem cell-based therapies. There are other types of stem cells in the body which have the ability to regenerate various different compartments. For example, hematopoietic stem cells are a stem cell type that can be used to regenerate the entire hematopoietic system. In terms of the actual stem cells that could recreate all of the cells of the human body, I think the field is still a bit mixed around the opinions on that. There have been, I think, some efforts to curb the use of editing and therapies in stem cells recently. So I think the field has to be very cautious when it comes to anything where true pluripotent stem cells are involved.
And when we develop autologous therapies, we're using adult hematopoietic stem cells, and we try to modify those cells, and then we then engraft them back in the body. Again, same for T cells. It's adult T cells, and so we're not developing any therapies or programs with germline or placental or saving cells. It's all adult systems.
Right. So we talked about the cell and gene therapies. I wanted to ask a bit about CRISPR. From what I understand, bacteria use CRISPR. It's a mechanism that bacteria will use to modify their own genetics to fight off disease, et cetera. Can you tell the audience what is CRISPR? How does it work? It's a new buzzword.
Sure.
In science, and what kind of work is being done by Novartis?
Sure. So it is a new buzzword, but in fact, the term CRISPR has been around for quite a while, since the early 2000s. It stands for clustered, regularly interspaced, short palindromic repeats. I know that's a mouthful. But really, as you mentioned, it was identified first in bacteria as kind of an interesting observation that bacterial genomes had these repetitive sequences within the genomes, and it was later identified that these repetitive sequences actually had similarity to the genomes of various viruses that infect bacteria. And so what bore out over the next 10 or so years was that this served as a kind of a primitive immune system for bacteria that allowed them to recognize foreign DNA that was coming in from an invading virus and to chop it up into little tiny pieces. A significant amount of effort went into this. Ultimately, this was determined to be a potential way of programming genome editing, that is, the induction of double-stranded breaks in DNA, in other types of systems. What's borne out now over time is that this is a very flexible system that allows us to induce double-stranded breaks into virtually any genome, to do so with a relatively high degree of specificity and efficiency. And at Novartis, we were very early adopters of this technology. In 2012, 2013, when some of the first papers were published demonstrating the utility of this technology as a potential genome editing strategy, we've invested a significant amount of resources in understanding the technology and being able to utilize this technology for both basic research as well, potentially, as a therapeutic modality.
So what kind of therapies are coming out of this? What's being worked on that you can disclose? What are the goals of some of the therapies right now?
We're developing CRISPR-based therapies for our lead indication is, again, an ex vivo modified autologous cell therapy for sickle cell disease. In that we take hematopoietic stem cells from sickle cell patients, or at least this will be our plan when we get there, to gene edit them with CRISPR-Cas9, and then to reinfuse them back into the conditioned patient. And that those modified hematopoietic stem cells will repopulate the hematopoietic system and then produce red blood cells, which have a different form of globin protein in the red cells that prevents the sickling of those cells and hopefully will be long-term and prevent the complications of sickle cell disease due to the sickling of the cells. And so we're in collaboration with Intellia Therapeutics and working closely with them to identify guide RNA molecules which target the region of the genome that will have the biology and will upregulate this alternative form of globin protein. And we're in the middle of preclinical evaluation right now on our way to the clinic. And so this is one example of where we're developing therapeutics using the CRISPR-Cas9 technology.
Well, because of CRISPR-Cas9 and how it works, can it ever be used in vivo, or it always has to be done outside the body in a controlled, safe environment?
Yeah, in theory, the technology can be used for in vivo delivery as well. We have opted to focus initially on these ex vivo cell therapies so that we can provide as much of a quality control over the final therapeutic product, which is the gene-modified cells, to really be certain that we have a favorable safety profile of these cells prior to being readministered back into a patient. There are several companies that have active programs around in vivo delivery of the components of the CRISPR-Cas9 system. So we're very excited to see how some of those in vivo strategies evolve over the coming year to several years.
Is there a trade-off between in vivo and ex vivo? Is there a reason you'd want to do in vivo, or is it always cleaner and simpler to just do stuff ex vivo and reintroduce?
Well, there are certain therapeutic indications where ex vivo really isn't an option. If you have a disease indication, like in this case CAR T therapy, where you're treating a tumor, you have the I'll call it the luxury, the ability to actually isolate a patient's T cells, genetically modify them, and then re-administer them back to the patient. The same can be said for the hematopoietic stem cell therapy that Sue just mentioned. Other diseases are not as amenable to an ex vivo approach to genetically modify the cells. So, for example, in some of the retinopathies, one would actually need to perform the gene editing events in the retinal pigment epithelial cells in the eye in order to see a therapeutic benefit, there isn't a strategy where those cells can be cultured ex vivo and then redelivered. So it really are the specifics of the disease indication that determine whether or not an ex vivo strategy is even possible.
Okay, that makes sense. And I guess in certain circumstances, you may need to be highly specific, both where in the body and what type of cells. Other circumstances, maybe it's more generic and it can be it can happen. Maybe you can inject something and the modified cells can travel to the proper site. So I guess it obviously depends on the therapy, but both techniques need to be used.
Yeah, that's right. It depends on the therapy, and it depends on the method by which you're delivering the particular cargoes to given cells. So there have been a lot of excitement in recent years with AAV vectors. AAV vectors do have the ability in some circumstances to transduce certain cell populations, but it's not exquisitely specific. So the Avexis SMA therapeutic that Sue was mentioning, this is one which is based on an AAV serotype that happens to have a very high efficiency of transducing cells of the brain and motor neurons, and that's one of the reasons why it's an effective therapy.
Okay. How do you see this field evolving over the next few years? Do you see an explosion of new therapies, or is it still going to be chugging along and there's still a ton more to figure out before we can get a lot more therapies out there? What's your overall perspective?
Well, I go back to earlier this summer, the FDA commissioner was interviewed, and he actually cited that he expects by 2022 that there would be 40 new approved gene therapy medicines on the market. He was citing an MIT study that looked at the ClinPharm pipeline from last year of all the programs that are in preclinical development and all the ones that are in the FDA under review as part of investigational new drug applications, and put on a certain metric as to success rate and attrition of the total and how they progress through the different phases of clinical development. And that's where he came up with this number of 40 new gene therapy medicines by 2022. So that's a really exciting for the entire field. So he's also working towards putting in accelerated programs to help with accelerated reviews for sponsors and teams to take advantage of, because they see that gene and cell therapies are becoming a reality, and there will be more on the market. And in fact, that's the excitement within Novartis and why we are making this a strategic growth area across multiple disease areas. So I think it's a very exciting time right now.
Yeah, no, that sounds like it. Yeah, I'm sure your ears are more finely tuned than mine. I'm sure you hear all the time of people having various diseases and there's no therapies available and then passing away. And so I'm sure everyone feels the urgency over there to work as fast as they can to get therapies out there.
Mm-hmm. Yes, absolutely.
Well, very good. So, we're just about out of time. What's the best way for listeners to find out more about what Novartis is working on and to reach out and ask questions?
So there's a wealth of information on Novartis's public internet site, Novartis.com. We're also very active in social media, especially Twitter. So I would encourage people to follow Novartis on Twitter. We are very active in giving updates around some of the new areas that we're investigating. And our global head of research, Dr. James Bradner, formerly from the Dana-Farber Cancer Institute, is a very active contributor to Twitter. And I would encourage people to follow Jay Bradner's Twitter feed.
Well, that's great. Well, Susan and Craig, thank you so much for taking the time. I really appreciate it.
Thank you very much.
Yeah, thank you.
You have been listening to Almost Here, Around the Corner Future Technology podcast with Richard Jacobs. Subscribe to this podcast, post a review to discover more future technologies that are poised to transform our lives for better or worse, such as Bitcoin, artificial intelligence, 3D printing, blockchain, virtual reality, and more.